Third team up is Team GI Exosome Capsule, and they are coming from the Anderson, Langer, and Sharp labs. Stage is all yours. Anybody need this?
I got this.
No, you have slides.
Yeah, got a clicker.
How you doing? So, do we have the slides?
Yeah.
Hi. So, welcome. I have the major role of introducing this, and this is a collaborative project between both Phil and Dan and my own lab, and the people who are doing the work are Gio, Suman, Amia, Farad, Natalia, Yulia, Vikash, Yunha, Sufang, Salil, and Makleva. And so what I just want to do is make a brief introduction. So the problem that we're going to deal with is colon cancer. And as people may know, especially older people like me, that you have to, if you're over 50, you usually have to go in to get a colonoscopy, and it's not the most fun procedure. And I say everybody's nodding, but actually maybe even more importantly, even in the United States, there's something like less than a third of the people that do it. So and that's one of the reasons why, it's such a terrible kind of cancer, because people do not do that detection. So Gio Traverso, who actually is a fantastic example of convergence, he got his PhD with Bert Vogelstein, and he's a card-carrying gastroenterologist who actually sees these patients all the time, and actually is doing some great work here in the Koch, is going to explain this to you a bit more, and then Makleva is going to go over it even further.
Great. Thanks so much, Bob. So, as Bob mentioned, I've actually been working on this for quite a while, close to 18 years, and I'm very passionate about this problem, both from the research perspective, but also from the clinical perspective. And just to give you a sense of the magnitude of the problem that we're talking about, GI cancer in the US basically will result in the diagnosis of the cancer in 7 individuals over the course of this 12-minute talk. And also, over the course of those 12 minutes, 4 people in the US will die from a GI cancer. And as a clinician who sees these patients every week, I find this extremely frustrating, because we recognize that we have the opportunity to mitigate the morbidity and mortality from this disease. And here we have some data from colon cancer, which is one of the best characterized GI cancers, demonstrating that if we identify these tumors early, and what you see here is that in stage 1 disease, we're able to reach survivals upwards of 90% when we're able to detect those tumors early. And actually, if we detect them even before their cancer, in their precancerous stages, we have the capacity to reach near 100% survival rates. And so, from a clinical perspective, I see patients, and I really try and encourage folks to really get screened. The challenges, though, and we have many tools for screening of GI cancers, and some of those tools include endoscopic techniques like colonoscopy, which you see up here, as well as some imaging modalities. And some of the challenges that we have with these current systems is that they are invasive. They have complications associated with them. So, for example, with colonoscopy, there's a risk of perforation and even death from colonoscopy. And with imaging modalities, there's even the requirement for bowel preparation. There can be allergies that are associated with the actual procedures. And so together with the invasive nature and the complications that are associated with these modalities, as well as the stigma that's associated with screening for these tumors, we have poor compliance, as Bob was saying, at the level of the population. And so what we are aiming to do, and what we've started doing, is developing the next generation of diagnostics for the detection of tumors across the entire GI tract. And this really aims to detect tumors at their earliest phases, whether they are in the esophagus, the pancreas, or the colon. And the way that we're approaching this is by looking at exosomes and leveraging some of the basic biology that has shed some light into a very unique opportunity from these vesicles that are budded from cells. And exosomes carry a host of different molecules, including nucleic acids like RNA and also proteins. But what's unique about them, and really what provides the opportunity here for early detection, is that they have a coat. They have a membrane. And that membrane protects those biomarkers inside that otherwise would not be accessible for early detection. And that is what we are detecting and capturing throughout the GI tract, identifying tumors at their very early phases of development.
So our team has a solution to non-invasively capture these exosomes from the GI tract. We envision an ingestible capsule, similar to your daily multivitamins. The big difference is that our capsule will not degrade or dissolve in your body. When you swallow the capsule, it traverses through your gastrointestinal tract and only begins capturing exosomes at a specific site, in this case, the colon. Then, the bound exosomes will be extracted from the excreted pill, and their RNA will be isolated for sequencing and screening of GI cancers. So, how does this actually work for you as a patient? Well, let's show you. Bob's gonna swallow our exosome capturing capsule. It doesn't exist yet. And now it's gonna pass. And he can actually, before the toilet flushes, he would have picked it up with a magnet, since the capsule will have embedded magnetic components. Using FedEx, Bob can ship his capsule overnight to the nearest sequencing center. At the sequencing center, they will open the package and extract the bound exosomes from the capsule and isolate Bob's RNA for sequencing. And in no time, Bob will have his RNA sequencing results on his famous smartphone. So what's in our exosome capturing capsule? We've designed our capsule with the outermost layer being an intestine-specific coating. So again, when you swallow this capsule, this coating will actually only dissolve when it reaches the colon. And now, this enables exosomes to enter this exosome filter. This filter enables separation of nano-sized exosomes from larger matter, such as fecal content and bacteria. Now, with exosomes flowing in through the filter, they can be bound and captured by the exosome's capture matrix, which is the innermost layer of our capsule. Therefore, these exosomes are protected in the device as it traverses through the rest of its journey in the GI tract. We've already begun looking for exosomes in intestinal fluid from pig tissue. Our preliminary data indicates successful isolation of exosomes. And we have taken those same exosomes and isolated RNA from them, and have good quality RNA for sequencing. This gives us confidence that we can isolate RNA from exosomes in GI fluid. During the first phase of this grant cycle, we will actually focus on assembling the various components of this capsule, as I mentioned before. And then in the second phase, we will evaluate the device's sensitivity, specificity, and utility. To test the safety and passage of the device in a large animal model, we will administer the capsule to healthy pigs. At the end of 1 year, we will have a capsule to non-invasively capture exosomes from the GI tract. The aging population in the United States is growing rapidly. In fact, today, there are more than 100 million people eligible for colon cancer screening. This presents a domestic market opportunity for our technology of over $4 billion. Our technology is the future of GI cancer screening. It is non-invasive, patient-friendly, and uses genomics as a diagnostic. Colon cancer is the least prevented, yet most preventable cancer, but compliance for current diagnostics is very low. We know that our technology will improve patient compliance for GI cancer screening while decreasing the cost. The current rate for colonoscopy is $4,000, while we estimate the price of our capsule to be less than $10. We can also adapt our capsule to detect other GI malignancies, such as small intestine cancer and pancreatic cancer, for which there are no currently available early diagnosis tools. We can also detect and monitor inflammatory bowel disease, which increases the likelihood for colon cancer. Therefore, our multidisciplinary team from the Langer, Anderson, and Sharp Lab hopes to use our exosome capturing capsule for both early prevention and detection of GI cancers. Thank you.
So first, I think it would Absolutely be huge if we could get earlier detection of colon cancer, and colonoscopies are certainly not a pleasant experience, although I haven't tried it myself, but I've read online that actually a bottle of vodka with the preparatory liquid could actually improve compliance with the procedure. So you might want to study that. Presumably, the number of normal exosomes is much bigger. So could you talk to us about, first of all, is this a looking for a needle in the haystack type problem? In which case, how likely is it that when you pass this capsule that you're actually going to capture relevant exosomes to study? And if there is a rarity of these types of particles that you're looking for, I understand that colorectal microRNA have been detected in plasma. So would it not just maybe be easier to do this by tests of other bodily fluids where you might be able to get larger volumes and actually have a higher likelihood of finding these?
Actually, it's a good question. So the problem for other bodily fluid is, for example, for blood, the exosome from the blood actually come from all the cells in the body. And because we only care about GI cancer right now, and so if we take the exosome from the blood, there's a problem for the dilution. So think about this: the blood the exosome is from all the cells in the body, which contains a lot of normal cell and a lot of cancer cell exosome too. So there's a ratio of the signal to the noise that the normal cell exosome will cover the real cancer cell exosome that we care about. But in contrast, if we only take the exosome from the GI tract, where the cancer exosome will be in the dominant ratio, then more likely we're going to figure out the biomarkers for the GI cancers in this case.
And if I can actually add, so a couple of things. So as far as the early detection of a tumor, so we actually showed in the New England Journal in 2002 that it's very difficult to detect early stage tumors, i.e. large adenomas, even in the feces. And you're right, it's a signal to noise problem, but what the exosome does is that it opens up the opportunity to basically use the central dogma, i.e. RNA as your amplification step. So instead of just having the 2 copies of the mutation, now you have actually amplification from the RNA. And so now your signal to noise is actually, we actually now have the chance of finding that needle in the haystack because we're now, we're protecting the RNA and we're querying it. So I think that's an important piece. And I think the other thing is, yes, we can detect in cancers, we can find RNA in the serum and DNA as well. But by the time that it's in the blood, it's a little late. And what we're really trying to do is capture tumors when they're on the luminal side, on the inside, before they actually progress and penetrate the vasculature.
So, and then you're going to really pass a screen through this, and you've got these little beads that have to accumulate material. What do you know about the ratio of the exosomes that you're looking for versus the ability? At what point do these screens become saturated, and is there a risk that these things become saturated before you find any relevant exosomes?
Well, in our early phase, we're going to do a lot of the tests. We're going to do the exosome capture technology on the normal cell versus only on the cancer cell, and we're going to identify this biomarker first. And also, we're going to do the sensitivity analysis based on the testing our device on a mixture of cancer cell and the healthy cell in different ratios. So we're going to test our sensitivity of our device specifically on these purposes.
Do you have
a? Just similar question to what I asked last time. Do you have in your write-up, you said that the current tests, non-invasive tests, are inadequate. So what is your goal for sensitivity and specificity in order to make it more adequate? And then I have a follow-up question once you answer that.
So I'm happy to take the question. So, I think that the answer of what sensitivity and specificity, we would actually engage with, number one, the payers to identify what sensitivity and specificity will help drive the reimbursement of the product here. And because I think recognizing the costs that are associated with poor specificity, but then also recognizing that lower specificity may actually translate into higher sensitivity. So I think identifying that optimum, but really engaging with the payers to identify what optimum. And I think when you have the entire transcriptome and expression as sort of, that you can query, I think the potential for really high both sensitivity and specificity is there, but at a cost. And I think identifying and doing the cost-benefit analysis in collaboration with the payers is what we would try to do.
So the follow-up, because we're short on time, the follow-up, it will take me longer to formulate it as a question, so I'll tell you kind of, whenever you get to be our age, so I was struck by Bob saying he doesn't like colonoscopies. 20 years ago, I didn't need a colonoscopy, and I thought I could find a way not to ever need one. And we started a company called Exact Science, which has this test today called Cologuard. There's two things you should know about it. One, it's got 92% sensitivity and 87% specificity, which is damn near a colonoscopy, and it's been very poor adoption. Second, the FDA approved it 20 years later. And so the thing you should do, though, is that exosomes, so what's the message out of that? One is, kind of the laudability of the desire doesn't necessarily get everybody to help you get it out. I think that the exosome part of this is kind of the most powerful thing. So if you could actually collect these exosomes, I think that you might find a lot more value, as you're hinting, in the actual kind of information richness you're going to get for therapy, for targets, for a lot of other things. I worry in a way, just as a question is, if you can't get headway in the 90s or very high 80s, really will there be enough proof that 98% is so much better that it's worth spending $100 million developing something? That's the question you're going to have to answer.
No, and I think the Exact Sciences lesson, and I said that I had been working for about 18 years, and all of the technologies I developed were licensed to Exact Sciences, so I know them well. And I know what they've done and their challenges they face, and I think recognizing and understanding them actually helps inform what we're doing. And I think what the exosome sort of opens up is the possibility really of a single capsule for all of GI cancers and several sort of precancer pathologies. And as you say, sort of really looking at the potential for some really high-level bioinformatics to, guide, basically treatment decisions, but then also early detection. So,
Thank you, Chris. If I could ask a question real quickly. The colon prep, it seems like part of the main commercial argument is you're going to avoid colon prep. I'm assuming, though, the sensitivity of the technology would be enhanced if there was colon prep. Is that right? In other words, it's like you're trying to avoid something, and in fact, almost to Nubar's point, which is there's maybe so much powerful information you're learning, and in fact, if you do colon prep, you may actually make it easier to collect that information, or is that not necessary?
So I think for the bowel preparation, it wouldn't be needed. And in fact, we probably, and I think we would have to do the clinical study to see which one essentially maximizes release of the exosome. I think that's an experimental question. But I think what we're aiming towards is actually not having the prep, because really what we want to do is engage with the entire population and make it really as accessible to everyone as possible.
Yeah.
All right. Thank you so much.
Thank you.